Hello, my name is Riley Anderson, and the title of my research project is The Effects of Sudden Cold Snaps on Honeybee Colony Function and How Rapid Temperature Drops Alter Brood Development and Worker Bee Activity in the Apis mellifera. The importance of Apis mellifera is that they are also known as the Western honeybee, and they play such a large role in pollinating all the crops we have all around the world. Um, the focus of this research is that the this presentation will focus on how the extreme temperatures can affect how the extreme temperatures can affect the Apis mellifera and the worker bees and their brood development itself. Um, we know that the threats to honeybees, they're so fragile and their hives that can be affected by just little things, and so we're going to see how this is a threat to them. My research question is, how do rapid temperature drops affect brood development and worker activity in the Apis mellifera, and what are the consequences for colony survival? This first experiment um, kept bees at a cold temperature for 36 hours. They specifically tested the fat body and its tissue. They found that there was a significant increase and decrease in metabolites. And then they tested, and then the significance of this is that they found that it changed the resilience of the bees, uh, and the fat body tissue altered the resilience. And so this meant um, that the resilience, once the resilience was changed later on in their life, if they had a cold snap, then they wouldn't be able to come back from it, therefore changing their long-term colony or therefore being a long-term colony effect because they just wouldn't be able to come back from this kind of thing. The next experiment was that they also kept them at a cold degree, 20 degrees Celsius, for up to 96 hours, starting at 12. And they specifically tested the mortality rate of brood and the misorientation inside their cells themselves. The key findings were that if the pupils didn't die because they had a very high mortality rate, then most of them would be misorientated. And then after they were misorientated, it led to worker bees emerging, or worker bees had significantly reduced longevity. And so when they're misorientated, it could have messed up something inside the cell in their exoskeleton, leading them to not be very healthy in the long term. And so they died quicker, even if they died quicker after they had emerged from their cell. This also led to nursing bees being forced to be foragers earlier in their life, which made them less efficient on food processing and gathering pollen, which was very bad for colony health and long-term colony health, which could lead to a colony failure, which is not very good. Here we can see on the top left, there's misorientation. In the bottom right, we can see just how large that gap is between honeybees that survived and the treatment and the control group here. Um, and the third peer-reviewed article, we have the experiment. They did a cold stress at 26 degrees Celsius for 24 hours, heat stress, and vero destructor stress. Here, the key finding was that the cold stress was the most impacted, kind of proving that how bad cold stress is for these honeybees. And that meant that they're premature. They left the hive prematurely. And when the significance of them leaving the hive prematurely is that these bees they weren't ready to go out, and so they weren't being efficient when it came to pollinization and bringing back anything to their hive, making them efficient. And so they actually didn't help out their hive because they were they were not being efficient at all. And this was a long-term effect and could also lead to colony failure. Um, and so that cold stress just didn't do anything to help, especially in the worker bees. And when the worker bees aren't working very good, it hinders the rest of bees, it hinders the rest of the whole colony. Here we can see that the gap between the cold and the cold control, how much higher the treatment was, or sorry, how much higher the stress was on them because they left prematurely. And our conclusion is that cold stress pre-pupa showed large changes in their metabolome, showing that uh, the cold stress environments, their tissue was affected. and it was affected negatively on the prepupa. The cold stress pupil also had an increased mortality rate, and the brood that did survive had a high chance of being misorientated, and after being misorientated, left them with a shorter lifespan. They also had the cold and heat stress, along with the viral parasitation, caused young worker bees to leave prematurely, with the cold sticking out in this experiment, showing that showing how negative cold snaps are towards honeybees, um, leaving them very highly, excuse me, leaving them highly impacted by the cold, and a negative standpoint there. And the significance of this is that cold stress has major long-term effects on individual worker bees and hive colonies. Cold snaps reduce strength in worker bees, and it especially reduces strength and creates a high mortality rate in the broods. And when cold basically targets the honeybees at such a early development rate, it can really mess up generations of honeybees. And when they don't target, and if they, excuse me, and they also target the worker bees, which is also the heart of the beehive, working for them. And so when the cold targets them, it really takes out the whole hive. And one thing that beekeepers can do is they can stay proactive when it comes to these weather changes. Insulating hives and positioning them away from the wind might help the cold, uh, might help the bees from staying away from the cold and not opening up any sort of hive when it is cold, because just a fraction of that cold snap can ruin a development of pupa. And the bee population is so important to human life, and pollinization is so important to the world, that we have to do everything we can to keep bees safe. Here are my references, and thank you for listening.